Sounds good.
Hello, everyone. Maybe we should try and get people in and sit down, that we can get started with the session. We're running over time, and so, yeah.
I think we'll just get started.
Okay. So welcome to the third session of this morning, which is on signal transduction and modeling in VHL disease. My name is Alessandra Boletta. I'm head of the cystic kidney disorders unit here at San Raffaele. And even if I'm not involved in VHL directly, I'm learning a lot because I think there's a lot of overlap between the disorders we study and this condition. And so it's a true honor to be here and share the chairing with Dr. Srinivasan, who will start and open the session.
Thank you, Alessandra. I'm Ram Srinivasan, a medical oncologist at the National Cancer Institute. Why don't we get started with the session? The first talk is going to be presented by Dr. Qing Zhang, a good friend and a colleague from the UT Southwestern. He's going to provide some new insights into VHL biology. And Qing, why don't you get started?
Thank you,
Looking
guys.
forward to your talk.
All right, thank you. Thank you for the invitation. It's really a great privilege to be here. So today, for this session, I'm going to start with introducing some new biology on VHL signaling access, especially in the setting of kidney cancer. So this is my disclosure. So at this moment, we're already pretty sure that VHL is most important tumor suppressor in kidney cancer. Obviously, actually, led by Tang, as well as the summarized a few years ago in this landmark TRACERx paper. But in terms of therapeutics, I want to quote Bill Kalin's, my old mentor, saying that some of the mutations that arise later in the revolution exist is because of the pre-existing mutation or truncal mutation preceding them. So if that's true, if you target those truncal mutations, potentially it can kill those tumors so selectively. So also actually give an analogy in terms of this Darwinian trait drawn by 1860s German scientist Ernest Haeckel. So you think about this Darwinian tree. This is bottom is the trunk, and this is branch. If you target those branch events, there's always going to be resistance developing. On the other hand, if you target those trunk events, you will most likely eliminate tumor cells growth. This is also actually happened to existing FDA-approved therapies, including ER, EGFR, BRAF, BCL-ABL, as well as AR signaling in different cancer settings. So those are successful examples lying in place. So today I would like to discuss a few potential strategies that we can target those truncal events in the case of the VHL loss in kidney cancer. I want to talk about briefly 4 things we could potentially do. So one is going downstream, such as targeting HIF-2alpha TKI, and then second is targets unique vulnerabilities in kidney cancer, such as lipid droplets. And third and fourth, I would like to talk in total together as a whole, because we can talk about synthetic lethality as well as PROTACs. And some of the colleagues here are also working on. So first, going downstream. So in terms of targeted therapy in kidney cancer, this is a nice review by Tony Shorey and Bill Kalin a couple years back in Nature Medicine, and summarized very nicely, coming from upstream HIF signaling, and then this will induce the downstream of the growth factors binding with respective receptors on the endothelial cells. And then previously, the targeted therapy against TKIs mainly involved multiple tyrosine kinase inhibitors that acting on VEGF, PDGFR, FGFR, or VEGFR receptors. On the other hand, we also know that belzutifan recently approved by FDA to treat either VHL disease or advanced kidney cancer. So those are remaining to be the main approach beside immunotherapy in kidney cancer. But obviously, I quote Marshall Nalahan, who originally cloned the VHL, this quote from 10 years ago, saying that, on the other hand, we're dealing with drug resistance, either intrinsically or adaptive resistance. Also, there's actually lack of response from half of the patients, some may be undergoing treatments. So the question is that we are probably ignoring other prospective CCRCC that may need to be targeted therapeutically to achieve maximal therapeutic efficacy. The burning question in the field at that time when I started in Bill's lab was, can we identify additional therapeutic target in RCC that is induced by VHL loss? Consider VHL E3 ligase targeting HIF. The burning question is, can we identify some other VHL E3-like substrates. And then the real million-dollar questions, because we spent $1 million on this, is how. So cut a long story short, this is after more than 10 years efforts with global efforts from various different institutions across the globe, from China, Singapore, Boston, Vanderbilt, North Carolina, Texas, and some of the investigators actually in this audience. So we actually identified some of the novel signaling axes through these global efforts over a decade. So in this case, I want to highlight some of the schematic we published previously after this long-term efforts led by two of the talented postdocs, former postdocs in my lab. Right now, they're actually running their own lab in China. So basically, we identified two of the novel VHL substrate besides HIF2-alpha. In this case, very little studies have been reported for those two proteins. Those are actually transcription factors. One is called SFMBT1. It's a polycomb complex protein. The other one is zinc finger homobox protein 2. In this case, one of the signaling axes through ZH2 is activating NF-kappa B, and the other SFMBT1 is activating PHK sphingoskinase 1, which is also later on collaborated by Ian Feruz's lab on this kidney cancer research as well. So those two studies showing that besides canonical HIF2-alpha, there's also the second and the third branch that is in parallel is activating this VHL loss signaling. And then by crippling those signaling pathways, we could potentially, in collaboration with HIF2-alpha, contributing to the kidney cancer tumorigenesis. Most likely, importantly, I would like to mention those HIF2-alpha resistant CCRC cells. If we actually inactivate SFMBT1 ZH2 pathways, we can actually slow the tumor growth and eliminate those primary tumor growth as well as metastases. So, and then second part I would like to really quickly touch upon is targeting unique biology of the kidney cancer, which we know that is clear cell renal cell carcinoma is majority of the kidney cancer, illustrated by this histology where you can see this big lipid droplet formation. And in the field, we know that this lipid droplet formation is really important to provide bioenergetics as well as dealing with ER stress in kidney cancer development. So the underlying mechanism still remains somewhat unclear. So in this case, by performing genetic screening, by collaborating with various investigators, many from UT Southwestern, we actually, using the genetic screening, and found that GM-GD6, which is TOG-dependent dioxygenase, can form actually complex with RBM39, which is transcription activator. By forming a transcription activated complexes, they can co-occupy those DGAT1 signaling along with H3K4 trimethylation marks. So DGAT enzyme is a very important critical enzyme that control the free fatty acid conversion to the triglycerides. And then triglyceride is obviously the component that actually induces the lipid droplet formation. From this perspective, DGAT1 represents the most oncogenic events that is controlling lipid droplets in CCRCC development. So once we figure out molecular mechanism, the most important thing is we can actually use DGAT1 inhibitors. In this case, we're using orthotopic models where we induce the tumor growth, and then in the control cells, after 5 weeks, tumor grow readily. But upon DGAT1 inhibitor treatment, you can see the slow of the tumor growth over time, suggesting this is one of the things we can potentially target in kidney cancer to eliminate the lipid droplet formation, as well as tumor genesis. The third and fourth thing I would like to emphasize there is actually coming hand in hand, because this regarding with synthetic lethality and as well as PROTACs. So this is the review paper we published recently showing that the concept of synthetic lethality, especially in renal cell carcinoma setting. So in normal cell, you could think about we have VHL wild type, which is gene that we often talk about, and then gene A will be the one that will be the synthetic lethality potential partners. In normal cell, if you inactivate gene A, in this case, because VHL is wild type, you don't cause lethality. On the other hand, because most of the CCRCC cells, we know that it's VHL now. So in this case, if you inactivate gene A in this setting, so you can see that actually they cause lethality. But the most important thing here is that because VHL wild type existing in normal cells, by targeting gene A specifically, you only cause lethality in the clear cell renal cell carcinoma. So to cut a long story short, we actually identified one of the important kinases, so-called TPK1, as a sensitive lethality pathway in kidney cancer. So TPK1, as we know from James Chen's lab at UT Southwestern, is a very important type 1 interferon response genes. When the organism sees the DNA, double-stranded DNA, they can sense by the CGAT signaling, therefore regulate STING activation as well as TPK1 phosphorylation on the downstream F3 pathways. The dimerization F3 pathway will induce the nuclear localization as well as activation of type 1 interferon response to actually fight back the double-stranded DNA, foreign DNA invasion in the tissues. But in this case, in a tumor intrinsic fashion, for example, we actually can deplete TPK1 using CRISPR-sgRNA. So we can appreciate in UMRC2, which is VHL now, it can cause specific lethality, but you restore the VHL in UMRC2 cells, you can see that those cells remain intact. And then this is also happening in another pair of cell lines we established. In this case, you can see specific targeting of those lethality in UMRC6 VHL now cells. The molecular mechanism through collaboration with Dr. Ken Gong and PKU, Beijing University, we actually found that VHL loss can induce hyperactivation of phospho-TPK1 and therefore induce P62 activation and leads to tumorigenesis. This actually corresponding with what Bill Caton's lab published, showing that P62, also called SQSTM1, is a pathogenic target of the 5q copy number gain in kidney cancer. So obvious question is, can we target it therapeutically? So through genetic model where we deplete TPK1 in an inducible fashion in an isotopic model, you can appreciate that those TPK1 depletion cause tumor regression, also showing quantitatively down there. More importantly, when we look at spontaneous non-metastasis, depletion of TPK1 cause totally abrogation of those non-metastases spontaneously. So in this case, we add extra layer of the signaling axis we're trying to target, which is phospho-TPK1 as well as TPK1 signaling. Obviously, compound 1 is a non-specific TPK1 inhibitor targeting IKK epsilon, which is not what we wanted. So we would like to develop a specific PROTAC that can be used to degrade TPK1. In this case, through collaboration with Lindsay Jens and UNC and Noel Williams at UT Southwestern, we indeed developed such PROTAC molecules, which is used to degrade TPK1 specifically in a dose-dependent manner. On the other hand, they wouldn't degrade the close family member, which is important for the immune signaling, so-called IKK epsilon. So we also can actually calculate DC50, which is showing about 100 nanomolar, showing very potent. On the other hand, the most important thing for PROTAC is actually specificity. In this case, we actually performed the global proteomics. In this case, you can see that TPK1 is one of the most abundant proteins that was being degraded upon the TPK1 PROTAC treatment. You might argue there's other couple proteins that can be degraded as well, but those turns out to be TPK1 targets, so as we validated experimentally. In this case, we're actually trying TPK1 PROTAC in animal experiments. So far, the data looks promising. So anyway, and then last part I would like to touch upon is the potential problem targeting TPK1, because we know that TPK1 is important for the multiple cellular activities, including CGAT-STING, which is important to mediate the cellular response according to double-strand DNA. But the more important question is, can we identify the specific oncogenic events that is activating TPK1 in autocrine fashion? In this case, we performed a candle-wide siRNA screening, basically using those 710 kinases, transfect CCRCC cells, and then did individual TPK1 phosphorylation, which indicate TPK1 activity, followed by the quantification of specific signals, and then plot those potential candidates out, and then eventually focus on individual candidate with individual validation in major kinase mass specs. The interesting part is that we identify a novel kinase, so-called DCLK2. So what is this kinase DCLK2? DCLK2 turns out is a neuronal kinase, which is important for the neuronal survival and the induced growth conformation. So this DCLK2 kinase has a kinase domain at the base, very C-terminal, but unfortunately, there's no kinase substrate being identified previously. In this case, we published a molecular cell paper recently showing that a various splice variant form of DCLK2 can specifically induce phosphatidyl phosphorylation, therefore induce PSH2 signaling as well as CCR6 tumorigenesis. So in summary, I would like to emphasize there's multiple signaling pathways which could be induced by VHL loss, which we can target therapeutically. But more importantly, what's ongoing research besides those signaling we have identified? We also work on some of the VHL-M6 regulation pathways. But more importantly, our lab has switched largely to using unbiased CRISPR screening to identify those potential novel targets. In this case, a study led by Qing Zhang, who was recently promoted assistant professor at UT Southwestern through collaboration with Celeste Salmon, Jim Berger-Wallace, and Srinivas Maladi. So we performed a genome-wide CRISPR-sRNA library screening where we infect those CCR6 cells with VHL now, subcutaneous inject into the mice, wait for 6 to 8 weeks, isolates lung metastases at primary tumors, followed by deep sequencing. So we can appreciate on 2 different cell lines, we can see concordance of the top genes that is being actually enriched in the lung metastases. So one of the genes we identified eventually is called HLF, mainly actually involving the hepatic leukemia factor in a leukemia setting. In this setting, we're using a topic model validated functionally, showing that CRISPR HLF knockouts can induce lung metastasis spontaneously while not affecting primary tumor growth. But on the other hand, overexpression HLF can actually induce this lung metastasis regression. So I know my time's just run out. In conclusion, I would like to emphasize 3 things. One is that we can target truncal mutation VHL loss. And second, we can use the sensitivity lethality. And third, CRISPR screening can help us identify some new therapeutic vulnerabilities, either in primary tumor growth or metastasis. So last, but not least, I want to thank all of the supports from collaborators or knowledge along the way, as well as my funding resources. I would like to take 1 or 2 questions if there's enough time. Thank you. Questions? Okay. Isama.
Thank you. It's a real tour de force of all this work. I'm wondering about the last bit that you showed with the regulator of metastasis. How is this implicated in tumors? Is it transcriptionally upregulated? What can you tell us a bit?
Yeah, that's a great question. Summer, this actually, I didn't have time to show this. This HRF is actually silenced, and then its silence contributing promotes this lung metastasis specifically, but not liver metastasis. We also have the CRISPR screening showing the other set of genes that regulate liver metastasis, in independent data sets. This gene silencing was controlled by the Swiss SNP complexes, for example, such as SMARC4. So in this case, we use a SMARC4 inhibitors. We can stabilize HRF, and then this stabilization will eliminate the lung metastasis downstream. So basically, it's a transcriptional regulation coming from upstream of the Swiss SNP complexes. And downstream is actually regulating integrin pathways with the transcription activity of HLF. Yeah, I'm happy to show more data for you next time when you visit UT Southwestern. Thank you.
Yeah, thanks.
Thanks, Qing. I mean, that was a lot of data that you presented. I'm going to focus my question on the DGAT1 story.
Right.
So can you tell us a little bit more about what inhibiting it actually does, other than, are there any metabolic implications to inhibiting DGAT beyond just altering, lipid droplet accumulation, 1, and 2, it looks like you have an inhibitor that goes after it. Are there any plans to develop it further?
Right.
How strongly do you feel that this is a good pathway to go after as a target in kidney cancer?
Ron, that's a great question. Always very insightful. I just want to quickly point out, DGAT1 has a family member, so-called DGAT2. So the previous publication already showed that DGAT1 is not an essential enzyme because you deplete DGAT1, mouse is totally fine. But DGAT2, on the other hand, is an essential kinase. Even though DGAT1, DGAT2 has similar names, they only share about 30% sequence homology. In this case, that's why it's very easy to identify a specific DGAT1 inhibitors, but not targeting DGAT2. So in this case, we argue DGAT1 inhibitor on its own will not have toxicities in normal tissues. given the reason that DGAT2 is an essential enzyme. In terms of your first question regarding DGAT1's function besides regulating lipid droplets, that's a great question. I'm not sure exactly how other signaling DGAT1 may mediate. The most reported or canonical function of DGAT1 is to control the lipid droplet formation as far as I know. But maybe there's a function not as aware of. But that's a great question. Thank you.
Thank you.
Can I add a quick one?
Yeah, of course.
Perhaps I missed it, but any of these, like TBK1, DGAT1 inhibitors, do they impact on HIF response at all?
TBK1 actually is totally HIF independent. So for example, if you deplete HIF2 alpha, you will not affect TBK1. Okay, and then TBK1 also not affecting HIF. So there's no, it's mutually exclusive. We're trying to combine TBK1 with HIF2 alpha inhibitors in those resistant cells, but most of the effect was actually mediated by TBK1 inhibitors. So, which means that TBK1 inhibitor plus HIF2 alpha inhibitor does not inhibit the TBK1 inhibitor's activity. But TBK1 itself, by either pharmacologic inhibition or depletion, will be sufficient to actually lead to the tumor growth regression. Yeah, that's a great question. Thank you.
Thank you. Thank you very much. So we move to the next speaker, who's going to be Ruhi Deer from the Baylor College. And she's going to talk about linking VHL and SETD2 in a common oncogenic pathway converging at the mitotic spindle.
Thank you. Hi, everyone. I'd like to begin by thanking the VHL for this opportunity to tell you a little bit about what's going on in the lab. We normally study chromatin-modifying enzymes outside of the nucleus in these new and novel functions that we're finding on the mitotic spindle. And today I'm going to give you just a small story where we think VHL and SETD2, which we know are moonlighting at the mitotic spindle, are in fact converging on a common pathway. So we've seen this model several times over the past couple of days, and essentially I just wanted to highlight that we have biallelic inactivation of VHL and loss of the 3P tumor suppressors, both in renal cell carcinoma and VHL disease. And the main question that we wanted to ask was, what exactly is the loss of these three chromatin modifying enzymes, the SETD2, PBRM1, and BAP1, really doing in addition to VHL loss? And to sort of give you a little history of what these enzymes do on chromatin, you have SETD2, and I'm going to start from the right and move towards the left. We have SETD2, which is a histone methyltransferase. It adds methyl marks on the histone tails. Here it's adding a methyl mark on H3, lysine 36. You have PBRM1, which is part of a larger complex called the PBAF complex, which is involved in nucleosome remodeling. And then you have BAP1 right here, which is a D-ubiquitinase. It removes the ubiquitin moiety from the H2A tail. But the one thing that I will mention here is that all of these enzymes, although I've just mentioned that they're chromatin modifying enzymes, they do not share a common etiology on chromatin. And what do I mean by that? They're not in the same place at the same time, okay? And why is this important? Because what we are observing is that there is this convergence of the 3P tumor suppressors at the mitotic spindle. What's known is that VHL was shown to localize to the microtubules. We've shown SETD2 as a writer. It lays the methyl mark on microtubules at lysine 40. We have PBRM1, which reads that methyl mark. And then you have BAP1, which is actually deubiquitinating gamma tubulin, which we won't get into today, but that's really interesting as well. And our goal in the lab was to sort of identify if there was a functional convergence, whether these enzymes, all localizing at the mitotic spindle, was actually happening for a specific reason. And you can imagine this is really important, because if we were able to identify and explore different mitotic vulnerabilities, this would be a great opportunity to sort of take these cells with the mitotic vulnerability and sort of just help them go towards mitotic catastrophe. And just to sort of step back and tell you a little bit about SED2 methylating microtubules, we were part of the group that made the original discovery of SED2 being a methyl transferase on microtubules. We have an antibody that was raised to this methyl mark, and what we see is that you see this beautiful localization or staining of methylation at the spindle poles here in the bottom row and at the mid-body. And the mid-body is nothing but a structure where the spindle microtubules collapse during cytokinesis, and that enables the separation of the two daughter cells. And if you lose SED2, you start seeing a lot of mitotic defects. Okay, you see a lot of lagging chromosomes, you see a lot of chromosome segregation defects, including chromatin bridges, and eventually you give rise to a lot of genomic instability, which is quantified as micronuclei here. But what's even more important and really exciting is that if you have monoallelic loss of SED2, which is what you would see with loss of 3P, you see that SED2 function is exclusively deficient for its cytoplasmic function. So you do not see methylation on microtubules, but it is perfectly capable of performing its chromatin function. So chromatin function remains unaltered. And so having told you that, I just told you that if you have mono or biallelic loss of SED2, you should have aberrant mitosis, and technically these cells should die. So the question then was, how do these cells that have lost 3P, how do they survive with just one copy of SED2? And to answer that question, we sort of moved to our next favorite protein, which is aurora kinase A, and I'll tell you why it's our favorite protein. But what's known is that aurora expression is high in multiple cancers, including clear cell renal cell carcinoma. And in fact, elevated aurora levels are associated with poor prognosis. And this is important because we had a previous publication where we actually showed that in VHL deficient cells, you have elevated aurora expression. And here is just sort of one blot where we had 786 O cells. These are clear cell renal cell carcinoma cell lines, where you see elevated aurora. And this elevated aurora can be rescued if you re-express VHL. Gosh, I'm trying to get this mouse to work. Where you re-express VHL, and of course, because it's a target for degradation, if you add MG132, which is a proteasome inhibitor, you should see rescue of levels, which is what we do. What we've also done more recently is using cells where we've got these cells actually from Dr. Genovese's lab at MD Anderson, and we also have a similar cell line from Dr. Ian Frew, where you have 3P loss, and we've tried to see what happens to aurora levels in these cells. And what I've shown you here is we've actually measured the fluorescence intensity of aurora at the mitotic spindle. And if you see here, we see a distinct increase in aurora levels, both by immunofluorescence, but also by Western blots in these cells that have 3P loss. And so the question that we wanted to ask was, could elevated aurora drive mitotic progression in these conditions where you have monoallelic loss of SED2? And why aurora kinase? So aurora kinase is a very important mitotic kinase. It is extremely critical for centrosome biology as well as for bipolar spindle formation. It's elevated across multiple cancers. And what I should mention here is that it's very tightly regulated. It's not just regulated temporally during mitosis, but it's also spatially regulated. And what do I mean by spatial regulation? So you have different pools of aurora within the cell. So targeting all of the aurora is going to be actually really bad for, in terms of toxicity. But what we're now realizing is that targeting certain portions or pools of aurora is going to be a far more precision medicine approach to regulating aurora levels in cells. And since it is an kinase, the first question we asked was whether aurora could phosphorylate SED2. And to do this, we used in vitro kinase assays where we essentially took recombinant protein, both for aurora and SED2. Using P32 labeled ATP, we see that SED2 was in fact phosphorylated by SED2. We've done the experiment where we've used an aurora inhibitor called MLN8237. It's also called alisertib, and we see a decrease in this phosphorylation. At the same time, we've also IP'd aurora from cells and used this in an in vitro kinase assay, and we also see phosphorylation of SED2. To determine the site that was phosphorylated by SETD2, we moved and did some mass spectrometry where we identified serine 2080 on SETD2 as a site that's phosphorylated. And we've generated an antibody which we use for immunoblotting. And here what I've shown you is just an increased expression of Aurora, GFP Aurora in cells, where we now see a nice increase in phosphorylation of SETD2. So the next question we wanted to ask is, what is the functional significance of this phosphorylated SETD2? What is it really doing in the cell, and why should we care? And so I just told you SETD2 is a dual function methyltransferase. It methylates chromatin and it methylates tubulin. And so we took these cells that we call here HEC-B9s. These are HEC-293 T cells where SETD2 has been removed. And so we've rescued these cells with either full-length SETD2 or S2080A mutant SETD2. And we looked at what happens to the histone methylation, and it seems like there's absolutely no change in H2K36 trimethylation. Okay? We then moved on to see what was going on with the tubulin methylation, and here in the lower panel you can see that when you have the mutant, you actually see less tubulin methylation. We then repeated this experiment in mouse embryonic fibroblasts, which had a tamoxifen-inducible system where we could get rid of the SETD2, and we've rescued these with either a truncated but fully functional SETD2 or a S2080A mutant. And what we see is that in the wild-type expressing cells, you don't see much of a difference in terms of tubulin methylation, but you see a distinct decrease in tubulin methylation in the mutants or mutant-expressing cells. So the next thing then was to see what's going on using microscopy at the mid-body and using the methyl antibody that we described before. And what we see is that in the S2080A, we actually see a reduction in methylation of the mid-body. Now, this is just fresh data that we got last week, and so we haven't quantified multiple experiments, which we're doing currently, but we do see a decrease in methylated tubulin. We also start seeing an increase in chromatin bridges, which is it's a chromosome segregation error. And since we were seeing this, we also wanted to see what happens to genomic instability in these cells. And we took this population of cells expressing either the wild type or the mutant SETD2, and we see an increase in micronuclear formation. But since this was a population, we also decided to go ahead and select clones, and what I've shown you here are 3 clones from the wild type expressing cells and 3 clones from the S2080A mutant cell lines. And hopefully you can appreciate that there's a lot more micronuclei in these cells with the mutant rescue, suggesting that this phosphorylation was in fact required for tubulin methylation and maintaining genomic stability. The other approach we've taken is using an Aurora inhibitor to see if that would prevent SETD2 methylation on microtubules. And here what we're seeing are human kidney cells where we've stained in green with the methyl antibody again. And what we're seeing here at very low doses of MLN, which is the Aurora inhibitor, we start seeing a reduction in the methylation of microtubules. Now, the reason we're using these extremely low doses of Aurora is because at high doses of Aurora, which is normally used traditionally in literature, we actually completely annihilate the normal mitosis. And so to prevent that artifact, we essentially use these low doses, and you can see a nice reduction in the methylation of microtubules, all the way up to about 200 nanomoles of the inhibitor. So what I've shown you in summary is that Aurora phosphorylates SETD2. I've shown you that its phosphorylation is required exclusively for microtubule methylation, but not so much for its chromatin function. And this phosphorylation also seems to be required for maintenance of genomic stability. So what we've now gone on to do is ask if phosphorylation of SETD2 is required for its microtubule methylation activity. Could we in fact inhibit Aurora and cause cells to undergo mitotic catastrophe? And so to do this, we set up a colony forming assay like Qing just showed you, where aurora inhibition was used again in extremely low doses with a SETD2 inhibitor. So in the recent past, we've had the good fortune of having a SETD2 inhibitor. And essentially, the second lane is with the SETD2 inhibitor alone. But then when we combine both the aurora inhibition with SETD2 inhibition, you start seeing a decrease in the ability to form colonies. This is true for A498 as well as the 786Os. These are VHL deficient cell lines, but they are proficient for SETD2. We've also done this in a genetically modified system. This was cell lines that we obtained from Dr. Ian Fru's lab, where he has A498s that are SETD2 proficient, and he's CRISPR'd out the SETD2. And again, we start seeing this decrease in the ability to form colonies. So this suggests that modulation of aurora is likely a good avenue to pursue. Now, I wouldn't say that we would take these aurora inhibitors directly to the clinic because, like I mentioned, they're extremely toxic, and there would be absolutely no one who would be treating patients with an aurora inhibitor. But I think this is really crucial because, as I mentioned before, aurora is present in multiple pools in the cell, and it is absolutely critical that we understand what function of aurora is actually being modulated by some of these epigenetic modifying enzymes. And what I have not shown you here is that NSD1, which is also present on 5q, is also modulating aurora in a slightly different way. And so hopefully next time when I come back, we can talk a little bit more about that. So I'll stop here and thank the team that's done all the work. Most of this work was driven by a postdoc, Manga Motrapu, who has been driving this work. We have fantastic collaborators both at Baylor as well as MD Anderson and University of Freiburg. And finally, thanking all of the funding sources for believing in this work and giving us an opportunity to try it. So thank you all, and I'll be happy to answer questions. Just on time.
A great talk and for being on time. So, there are any questions? John.
Very quick question. Do they have those SETD2 phosphorylation mutation in patients? I just wonder.
No.
No. Okay. So it's probably driving from the current aurora kinase activity.
Yeah.
Okay.
So SETD2 is lost in VHL deficient tumors. So you are blocking aurora, and then you are saying that cells don't like it. So what are the targets of aurora which are important for survival then?
That is a good question. So, we want to look at the earlier events, right? We want to look before you've lost both copies of SETD2. Right, because that was sort of the goal of trying to understand how these cells are actually surviving. But aurora, of course, has multiple targets, right? So SETD2 is not going to be the only target that aurora has. And so I think for this study, the goal was just to see what happens in 3P loss, and that's the reason why we moved to that. But you're absolutely right. There will be many, many other targets that aurora will be hitting.
Thank you.
Richard, no question? Maybe I can ask one that I suppose was in line with Rachel's question. Any role for primary cilia and basal body staining, location of SETD2?
So just to make sure that I don't just jump, because the moment I hear primary cilia, I get very excited. Yes, there is a role for primary cilia. I mean SETD2 at the primary cilium, which we've not discussed. But as Rachel pointed out, apparently I'm wearing a shirt that has primary cilia all over it. So if anybody wants to talk more about cilia, I'm always welcome.
Excellent. Thank you so much.
Thank you, Rohit. I invite our next speaker to the podium, Dr. Sakari Vanharanta from the University of Finland. He'll be talking about transcriptional control of renal carcinogenesis. Dr. Vanharanta.
Okay, thanks so much. Thanks so much for the invitation. It's been a fantastic meeting. And I come from the University of Helsinki, and my lab is really a cancer biology lab, but we use renal cancer as a model for many of the reasons that have been highlighted throughout the meeting. Just like Sandra said yesterday, that this is a great context to study tumor evolution. I think it's also a great context to try to understand basic mechanisms of cancer biology. And so here we have a very, very reductionist view of cancer, where there's a normal tissue, eventually gets a mutant cell, and then something happens, it becomes carcinoma, and then metastases. And in the context of kidney cancer, I think it's been really well established that it's a VHL null cell that initiates this process. And despite all these developments, there are a lot of open questions here. One of them is, for example, why is it that this leads to cancer in some tissues but not in others? For example, colon cancers don't have these mutations. Why does it take so long for this process to happen? And then how come some of these tumors eventually become metastatic and others do not? And so it's in this context of VHL null oncogenic signaling that we try to understand how cancers develop and progress. And the way we kind of think about this is that this is a lot of this happens because of control of transcription. And one reason to think like that is that the tissues are defined by tissue-specific programs, defined by our DNA, and that leads to tissue-specific transcriptional programs. And it's in this context that the mutations happen, and I think this is one reason why these processes are very tissue-specific. Okay, so essentially we have a transcription factor network that then gets perturbed by a mutation, and again, in this context, we know that it's the HIF signal. This is a simplified version. I am aware of HIF1 and all that, but in this context, it really is HIF2 that then eventually somehow changes this program, and then somehow this leads to metastatic tumors sometimes. Okay, so I kind of entered this field from the end through work where we try to understand how advanced renal cancers sometimes become metastatic, and we try to understand, we developed systems where we can study this, and we identified the mechanisms where the VHL-HIF2 pathway actually changes its output through enhanced co-option or hijacking, and this is an example of that. So the CXCF4 is a gene that promotes metastatic fitness, and we could identify this kind of specific enhancer that is regulated by NF-kappa B, and in collaboration with the HIF2, a dependent enhancer then regulates metastasis, and we've published this, and there's some details there. We've also studied the links between the tissue-specific transcriptional networks and the HIF2 signal, and try to understand whether their interaction would somehow explain, at least partially, why these processes are tissue-specific. Here we have identified, again, another enhancer-level interaction where there's a PAX8, which is renal lineage factor, and HIF2 bind at the same enhancer, which are upstream of cyclin D1. And this is important for cyclin D1 expression. And it happens so that there's actually a variant. So this chromosome 11 locus has been linked to increased renal cancer risk overall. And there's a protective value that seems to disrupt this PAX8 binding site here. And we've, again, experimentally tested this. And a prediction that arose from this work was that this risk is actually linked to ccRCC and not papillary RCC. And so we collaborated with people, with Stephen Chanak's group at NCI, I think, and tested this specifically, and we could actually see quite nicely that the SNP, which affects PAX8 binding, is associated with ccRCC and not papillary. Again, this is work published, but these are examples of how we can use really this pathway to study mechanisms of tumor development, and hopefully by doing this more and understanding better how these tumors develop, there might be also opportunities for intervention. Okay, so but now I'm going to talk about a slightly, again, different question related to the same thing, and this is trying to address the question of why does it take so long for these mutant cells to develop cancer. So, and this came about this work from this mouse model that was developed by our colleagues at Cambridge at the time when I was there still, Athena Matagido, who made a mouse model where they disrupted PPRN1 and VHL, and they found that eventually these mice developed tumors, and this is what has been shown by others as well, but there's a long delay in this process. And what we wanted to simply ask, what happens during this time when there supposedly is silence, essentially, phenotypically, but obviously something happens because the mutant cells are there, and we know because VHL disrupts the transcriptional control mechanism, so that we should be able to identify the cells right from the beginning and phenotypically track those over time. And we did this by single-cell RNA sequencing and also ATAC sequencing by just sampling these mice at different time points where we expected them to be kind of phenotypically more or less normal. And what we see here is just a UMAP where we have the proximal epithelial cells, S1, S2, and S3. These are the normal ones. And what we could find that all these different segments produced knockout cells. But through a careful inspection, we found that these were only the S1-derived cells, knockout 1 cells, that seems to progress. And what we saw is we see this HIF upregulated population right from the beginning, which is expected, but nothing really much happened for a long time. But only at several months later, most prominently at 20 months later, we see some sort of progression. Okay, and we did all kinds of analysis to try to understand what this could mean in practice. And obviously this is transcriptional data, so we can look at the signatures and genes and try to understand what that could mean in practice. There was two trajectories. The first one went down from this origin. We termed this population origin, and this was there right from the beginning. And then there's this other trajectory that then ends up going to this endpoint, which is endpoint of this model. This is not necessarily very advanced, but it's still something that has started to move from the VHL null cells that initially didn't do anything. So looking at the transcriptomics, what it seemed that is this first transition from origin to transition is associated with very strong upregulation of transcriptional responses, and then there's further modulation of that when we move down towards the endpoint. The first one, transition is also very strongly associated with the AP1 factor activation, and we could also look at correlation with clinical samples and show that it's the endpoint that then starts to transcriptionally globally a little bit resemble cancers. They are not cancers necessarily, but they're still progressing from the normal. Interestingly, there were a few hits here that kind of caught our eye. KLF6, for example, we had through a completely different line of research identified as a factor that promotes renal cancer growth. And actually, there's an enhancer here that is highlighted, I think, I can't really see, but it's also dependent on PAX8 and HIF2. And so this kind of fits with the idea that some of these processes are tissue-specifically encoded. And also, another one was that there's this KIM1 is upregulated here at the endpoint, which is a marker of kidney cancer that can be also even used in the clinic. So this was quite encouraging. But this also these signatures made us think of a field that we had not really talked about or thought about in the context of kidney cancer before, which was the field of renal injury, which is obviously well developed and clinically important on its own right. But this is an experimental thing or approach that's been used in the field quite a lot, where people have clamped the renal artery and then release that to cause damage and then transcriptionally profile these cells for. And there's multiple papers that looked at this, and there's these waves of transcriptional responses that then initially change the injured cells and then allow them to come back, whereas if there's too much of damage, the injury programs fail. And this was interesting because when we look at the transcriptional response, these are some of the examples. This is not our data, but our analysis from this paper. So there's really these waves, this massive upregulation of these AP1 factors and others, KLF6, and then this second wave that comes later, which is, for example, marked by SOX9. Okay, so this was interesting for this reason. When we go back to this trajectory and we look at some of those marker genes of these, they actually behave very similarly. So they get induced in this transition 1. But unlike in the injury models, they keep going up towards the endpoint. And then genes like SOX9 are only upregulated in the endpoint. So this was clearly a parallel. It looked very similar, but it's also at the same time somehow different. But these are also the mutant cells, so this kind of makes sense. And we've looked at this in multiple different data sets. This is another data set of renal injury. The top ones here, the cells are healthy cells, these are injured. And this is just a signature that marks the first wave of injury. And this is an signature that marks the failed type of response. But when you look at our endpoint signature, it's kind of marking both the early injured and the late or failed repair. So this again just goes to show that this is the same program, sort of the same program, but at the same time also not the same. So it's somehow perturbed. So we've now then tried to understand whether this is functionally relevant and try to correlate further into clinical data sets. Also, we've got some help from Ian Fru's team, who has a collection of different mouse models of renal cancer. And obviously, these are things that are hard to study in humans because most sporadic CCRCCs are diagnosed late, and obviously the VHL syndrome is a context where we can see these early lesions, but this is actually from the mouse model. And again, we see the same what we saw. We have SOX9 expressing early lesions, and they also have this KIM1 marker up. And then we have looked at human data sets, and they all fits. This is actually from a VHL patient, our Cambridge colleagues, so this is normal. There's hardly any SOX9, but this is an early stage CCRCC, and they have SOX9 activated. So there's clearly evidence from the mouse models and of all kinds of different mouse models and human data sets that this pathway is active in cancer. This is actually a metastasis model that we had used earlier to understand the metastatic processes. They also express SOX9. We can knock it out by CRISPR, and we can see clearly phenotypes in mice, and we have also overexpression studies and things like that. So it seems that this pathway is functionally important for tumor formation. So I guess that's, well, I think all the, oh, one more point actually. So one thing that we noticed also through sort of control experiments, because we have followed the mice for a long time, we wanted to look at also what happens in the normal non-recombined renal epithelial cells. And actually what we found is that some of the same markers go up also over time in aged renal epithelium. And there's this data by Yang et al. From Science, this is obviously, again, hard to have different ages from humans, but this is public single-cell RNA sequencing data. And it seems that these young kidneys don't have as much activation, whereas then if you go to older individuals, you start seeing more cells that have these markers up. And I think this kind of links these signatures, the injury and the tumorigenic processes and age in a way that I think suggests a model. And I think this is maybe not I can't say that this is a summary of what we have, but I think rather like a hypothesis that we should explore and the implications of this. Because we think about this in a cartoon, we have the normal epithelium, and that sometimes gets injury, and you could think about all kinds of risk factors and things like that that might cause renal injury. Most of the time, perhaps it gets resolved and there's no major consequence, but it could be that when you have mutations, that affects the way this injury gets resolved, and perhaps that has something to do with how cancer develops, how perhaps the risk factors are associated with mutations, and how that could lead to cancer. So I think this is more like an idea that I think we should explore in this context going forward. Okay, so with that, I think I'd like to just conclude that, so from the earlier projects that I actually just kind of introduce as very, very, very shortly as examples, it's quite clear that the effects of the mutation, they do evolve over time. And it's clear that the first clones that have lost VHL, the mutation doesn't have the same effect in those cells than it has in advanced cancers. And there's some clear examples, and we kind of tend to go quite deep and characterize specific examples as kind of conceptual kind of examples quite carefully. And so we have evidence that it happens at the metastatic site. So basically some metastasis genes are clearly activated by the tumor-initiating mutations, but there's been epigenetic remodeling that allow that. And also, we've linked this to tissue-specific programs, which I think are quite interesting. As examples, now PAX8 is expressed in other tissues as well, not just in the kidney. So this is not explaining everything, but there's clear and I think quite strong genetic evidence that PAX8, at least in this one locus, affects the interaction with, or is interacting with the HIF2 pathway, and that I think contributes to the tissue-specific nature of this. How about these other tumor types in the VHL syndrome? We don't know, but I think the same concept could quite well be true there. And then this second part, I think, links this kind of aging and tissue repair processes to the kind of mechanisms that then drive or allow the mutant cells to progress. And here I think there's quite a lot of open questions. We're trying to address some of those in this project, but I'm sure there's still plenty left. And I think this could be interesting, especially in the context of inherited mutations, and there's been a lot of discussion of how to perhaps prevent that eventually, and to understand the mechanisms that allow some clones to develop. I think is something that we should try to think about quite carefully. So I think this is all I wanted to say, just to thank the lab and the funding agencies and many, many collaborators that we have the privilege to work with. So thanks so much for your attention, and I'd like to take some questions.
we've time for a couple of questions. Lucrato.
Great talk. I'm intrigued by the repair process of tubular injury and the SOX9 rule. Have you, because there is a switch between SOX9 off and on in terms of tissue repair, have you actually examined this further in tumor progression?
I think that's a great question. We have not yet. So this is a very slow model. So it kind of makes it a little bit hard to work with it. But I think this is exactly a good question. And we don't know, but I'm kind of guessing that somehow the SOX off bit kind of might be inhibited by the mutations at least sometimes, and perhaps that could be. It doesn't even have to be a complete inhibition. It could be just the probability of that reversal not happening gets less, and that will eventually allow. And this is the kind of thinking that I'm kind of entertaining, at least, that it doesn't have to be all or nothing, but rather a change in the probabilities of these mutant cells ending up somewhere or not.
Thank you.
Hi, this is very interesting and kind of insightful, but did you have a more kind of understanding of the injury response based on the genes that you presented? Like, is the injury response involve kind of ER response? Is it involving DNA repair response? I mean, what is, and the reason I'm asking these questions is, can you tailor it, this is what you're going to do, to pharmacologic intervention?
I think these are excellent questions. I think there's a lot of genes there, and I think what is remarkable that this signature is very, very similar to what you see in all these other studies that have just done this hypoxic kind of ischemia reperfusion injury models in this, and these multiple papers that show this very similar data sets. And there's also kind of drug-induced damage. That response is very similar, but I don't think it's known what induces that. And so, again, in the mouse model is something that's possible to study, but it's quite challenging. We're trying to set up these kind of experiments now, but I cannot answer that yet. But there's a lot of things are going on, so I think there could be definitely possibilities for that in the future.
So one more thing.
Yeah.
To what degree this gene expression profile of the injury that you see in the double knockout correlates with just a simple HIF2 or HIF1 or a hypoxia signature?
No, I don't think so. I'm not maybe sure I understand exactly. Do you mean like in established CCLCC data sets, do we see correlation between HIF2 induction and injury?
A HIF2 alpha signature,
Right.
to what degree correlates with your double knockout transition signature?
Right, yes, it does. Yeah, so it is present in the endpoint, and it's of course mixed then in with other injury signatures. So we see the HIF signature already before we see the injury signature, but then at the endpoint those are mixed. And then we also see this failed repair injury model at the end. So it is something that is not quite the physiological response. It is somehow different, and we're trying to understand why.
So I just have two quick questions. One is about is in the context of familial renal cell carcinoma entities, would this be analogous in, for example, tuberous sclerosis or other symbols? Can we draw a larger context from that, firstly?
I think the data could well be in agreement with that idea. So because this is not in any way specified by the VHL mutations, but this seems to be like an epithelial injury response. We see, and Ian's mouse models also have different genotypes, but they also show the same thing. So I definitely think this could be a possibility and something to keep in mind going forward. Yeah, absolutely.
And with regard to epithelial injury, often that's accompanied with a fibrotic response. Have you measured a fibrotic upregulation of any flavor?
We have not looked at that yet, but I think that's a great question.
Thank you.
Thank you very much, Sakari. That was a great talk.
Okay, thanks.
We need to move on. The next speaker is going to be Gian Nicola Genovese from the MD Anderson in Houston. And he will tell us about cross-species functionalization of the cancer genome.
how do I go forward with the slides? It's just with the mouse?
Yes, the left one moves.
Whoops. Nope. Sorry. Okay, thank you. So first of all, I want to thank the organizer for inviting me. This has been really a great learning experience. I am still relatively new to the field. So just to give you a little bit of background, my laboratory is interested in understanding how solid tumors evolve, and we do so by using an in vivo cross-species functional genomic approach, mostly leveraging the genetically engineered mouse as a model system. So today I want to share with you some of the work that has been done by a very talented postdoc in my group, Luigi Perelli. So when he joined the lab, he basically asked a very basic question, which is specifically in RCC, renal cell carcinoma, which, as you know, is a usually relatively indolent tumor. What drives the emergence of clones with metastatic features? And although we have a very deep understanding of the genomic landscape of disease progression, this is also thanks to the seminal work from Samra, the TracerX, Charlie Swandon, and other people, we, at least at the time, we were lacking a model system, an experimental model that would recapitulate the evolution and the biology of advanced and metastatic disease. So, as you know, renal cancer are extremely heterogeneous from a clinical, pathological standpoint and genetic standpoint. But I think one common theme is the fact that those tumors are driven by inactivation of several tumor suppressor genes, many of those on chromosome 3p. So those genes are often embryonically lethal, or they really mess up the function of the organ when you conditionally inactivate them in the kidney or in other organs. So making a mouse model of renal cancer has been challenging. So to address this question, we elected to use a CRISPR-Cas9 based somatic mosaic editing approach. So basically we crossed a tissue-specific recombinase here under the PAX8 promoter with a conditional Cas9 allele. So we basically activate the Cas9 during embryogenesis across the entire nephron, and the reporter system, which is activated by sequential exposure to a Cre and a flip recombinase. So now we basically transduce this animal orthotopically by surgery or systemically with polycistronic adeno-associated viral constructs carrying packages of guide RNA against those genes or genomic region of interest if you want to do more complex chromosomal engineering, along with a latent flip recombinase that can be activated by Cre-mediated inversion of this cassette. So here basically the Cre recombinase activates the Cas9 in the nephron, removes the first stopper flanked by LOXP site in the tomato reporter, and then only in the cells transduced by the virus activates the flip recombinase. Now the flip recombinase removes the second stopper from the tomato reporter, which is flanked by the FRET site. So it basically allows you to track the fate of the cells that you're editing. So last 5 years, we tried a lot of genetic combination. We were able to model many tumor types. Here I'm reporting a model of a VHL-driven clear cell renal cell carcinoma. You can see the hallmarks of the disease, the prominent neoangiogenesis, both at the macroscopic level and at the microscopic level here by CD31 staining, the expression of the lineage marker PAX8, and then if you look at the classical H&E slide, you will see the prototypical cytoplasmic clearing, both in low-grade and high-grade tumors. But I think the most important piece of information that we gained from this model was the fact that if you disrupt the cell cycle checkpoint through genetic engineering of a genomic region on the mouse chromosome 4 that is synthetic to 9p, so basically taking down CDKN2A and CDKN2B, now we're really capable of inducing tumors that are not only locally invasive, but also metastatic. And the pattern of metastatic spread you can see is pretty similar to aggressive human disease with splanchnic metastasis, lung metastasis, and we also had 10% of mice developing brain metastasis. Histopathologically, those tumors are poorly differentiated. We see a lot of sarcomatoid or rhabdoid dedifferentiation. So this work was published last year, and we basically were able to demonstrate that the progression to aggressive disease is associated with emergence of chromosomal instability and disruption of the interferon response to as an adaptive mechanism to chromosomal instability, to tolerate chromosomal instability. So then we wanted to ask if there are specific vulnerabilities that are specific to highly aggressive disease. So we generated highly metastatic and low metastatic clones that we called M+ and N-, and we performed a genome-wide CRISPR-Cas9 screen. And to our surprise, running pathway analysis on the top candidate targets emerging from this screen, we found that M+ highly metastatic tumors are highly reliant on oxidative phosphorylation and complex 1, mitochondrial complex 1. This was true both in VHL-driven and non-VHL-driven tumors, which, for us was counterintuitive. So for a period of time, we were kind of stuck on those data. Then I have to say this was very fortuitous. Thanks to Jim Brugarolas, we got in touch with Ralph De Berardini's lab. So Divya, in his lab, basically did a very elegant experiment. So they took patients with renal cancer, and they infused those patients before surgery with heavy carbon-labeled glucose. And then we tracked the fate of glucose in the TCA cycle by mass spectrometry at primary and metastatic site. And what they found is very interesting and in line with our findings is that the incorporation of carbons derived from glucose in the TCA cycle is increased at metastatic sites, and that metastatic lesions are more reliant on oxidative phosphorylation. Now, if you look at transcriptomic data, you would also notice that, especially in advanced stage tumor, the presence of an OXPHOS signature of a signature of high mitochondrial DNA is associated with poor prognosis and is associated with high tumor grade. So, I also wanted to mention that in my lab, we actually validated these through spatial transcriptomic sequencing. And we looked at matched primary and metastatic site, and indeed we found overall at metastatic site a suppression of the IF signaling signature and an activation of an OXPHOS signature. So these are all consistent with previous results. So we joined forces with the Ralph group, and we designed some functional experiments. We used both pharmacological and genetic tools to suppress the function of complex 1. We did that pharmacologically in M+ highly metastatic clones. And indeed, in that case, we noticed almost a complete suppression of metastatic progression and activation of complex 1, hyperactivation of complex 1 in poorly metastatic clones. We did that by overexpressing East NADH dehydrogenase, which is extremely potent. And both in non-VHL-driven and VHL-driven tumors, we noticed a profound suppression of metastasis when you block complex 1. And this was associated with a decrease in cell proliferation. So this work was published in August this year, and now we can add a new piece to the puzzle where we basically know that the progression from poorly metastatic or non-metastatic disease to highly metastatic aggressive disease is associated with a metabolic rewiring towards oxidative phosphorylation. Still have time. So now I want to share with you a little bit of a different story. Just letting you know this is very preliminary, so please take it with a grain of salt. Here we basically wanted to study tumor predisposition syndrome using induced pluripotent stem cells, and you heard about this approach by other investigators yesterday. So the question here is, what drives the tissue specificity of tumor predisposition syndrome susceptibilities? Why some mutations, like the one that we observe in the Li-Fraumeni syndrome, are more pleiotropic, affecting virtually any organ with different tumor types, mesenchymal versus epithelial? And why in other conditions you see very specific tumor affecting very specific organs? So to study that, we used iPSCs. So we basically collected blood from peripheral blood from patients with genetic syndrome. We collaborated with the GU Oncology Clinic, with the breast oncology, GI oncology, and also with the pediatric clinic now. So we established the iPSC, and then we performed teratoma assays. And the idea here is to use digital pathology, spatial genomics, spatial transcriptomics to look how the mutations are affecting the tissue commitment within a teratoma. So this is what you usually see in a teratoma. You see tissues derived from the three different germ layers. This is from an healthy control. To be honest, I had to roll up my sleeve to provide the blood for these. You can probably appreciate the typical Southern Italian pigmentation here. But more importantly, at this point, we were able to generate iPSCs from many types of patients with cancer predisposition syndromes. And just to give you a little bit of preliminary data, again, take them with a grain of salt. Here I'm showing a teratoma generated from a BAP1 syndromic patient. We see a very strong bias towards neuroectodermal and neural crest differentiation. Here you have another set of teratomas from a VHL patient. This is a C393A mutation. Most of the teratomas were cystic in nature. However, in a couple of them, like this M2 here, we saw somatic transformation towards a neuroendocrine phenotype. You can see the expression, the staining with chromogranin A here. And very interestingly, this patient had an history of a pancreatic neuroendocrine tumor. Now, it doesn't mean anything, but for me it was quite interesting to see this phenotype. So the hypothesis that we're testing here is to basically look at how different type of second hits on VHL or other genes for other syndromes are affecting the lineage specificity, the lineage specification in those teratomas. And with this, I want to leave some time for questions. I want to thank the people in my lab. They're a very energetic group, collaborators at MD Anderson and across the institutions, and the funding agency. And I'm happy to take questions.
Super exciting, Gian Nicola. Are there any questions?
Gianni, that's really fabulous, all of it. In terms of the metabolic rewiring and the metastatic phenotype, and I'm probably being oversimplistic, but given that you've got CDKN2A as the driver there and its role in cell cycle progression, could this just be downstream of proliferative phenotype? And for example, in Scott Lowe's model, do you see the same metabolic switch, or is this really specific to VHL?
That's a very good. I don't think it's specific to VHL. What I think is that is more specific to chromosomal instability. So when a tumor develops high degree of chromosomal instability, we see this higher dependency towards OXPHOS.
It could be cell division dependent as well. I mean, aneuploidy also drives...
Potentially. So to be honest, we haven't really
yeah.
looked at the mechanism of this, switch.
And a quick second question, because with your teratoma assay, you said that you're looking for investigating how the nature of the second hit drives different phenotypes. So do engineer it before you implant these, or you put them in as heads and then you just wait for...
We thought about engineering, but
yeah.
we're just going to be with the spontaneous model. So whatever comes out spontaneously, and then we characterize with spatial genomics.
Yeah. Thank you.
Thanks, Amr.
Did you investigate what do tumors want out of OXPHOS? Is it ATP? Is it NAD+?
Good point. I think it's just more food. They really need more, as you said, more ATP, more reductive potential. But to be honest, this is probably a question for Ralph than for myself. I'm not like that great of a metabolic person.
Thank you.
Hi, did Ralph look for glutamine contribution?
No, I don't think they, we didn't do it, and I don't think they did it. I don't know if they're doing it.
Because essentially the whole thing can be explained with more HIF activity.
Say again?
The whole thing can be explained with more HIF activity, because HIF would drive OXPHOS, and HIF will drive also glutamine.
Yeah, that's, I mean, it's...
reductive carboxylation.
Yeah.
So it may all be compatible with the simple fact that when you go from primary to metastatic, your HIF levels go high.
That's very possible.
Yeah.
On a more philosophical standpoint,
Yes.
we noticed across cancer type, because we also work on pancreatic, melanoma, other tumor, when tumor advance, they usually tend to lose some dependency from the driver initiating oncogene. And I also think that this is happening somehow in renal cancer. So they might be, I don't want to be blasphemous here, but they might be less dependent on the HIF signaling.
Well, in the humans, they do accumulate mutations. We know this. Mm-hmm. So there is a possibility that they're becoming partially independent of the HIF. But,
yeah.
You know, in the setting of the animal model that you are testing it, with a specific inactivation of the pathways, I mean, you don't really know if your metastatic disease is primary tumor phenotypically independent. The only thing you know is more OXPHOS.
Yeah. No, you're right. And the truth is that we didn't look at the mechanism. So, you know, I'm really waving my hands here. But yeah, this is a very interesting point to address, absolutely. Thanks.
Just a kind of an open question here about the regulation of tissue specificity in VHL disease. Fascinating question, absolutely. Do you think that miRNAs regulating HIF2 mRNA might be relevant to this whole story, or is that something that I haven't heard anyone talk about mRNAs in this particular meeting, and I'm curious if that's even a topic that people are investigating.
I mean, yes, totally possible. We just don't know, and I will be open to any mechanism of regulation.
VHL loss wouldn't be relevant in tissues if HIF2 isn't regulated.
Yeah, that's pretty. Yeah, it's a fair point.
Thank you. I invite our next speaker, Dr. Ian Frew from the University of Freiburg. He's going to be talking about sex-specific differences in CCR/CC, separating the functions of KDM5C and KDM5D.
Thank you very much. Good afternoon, everyone, from my side. When I was trying to decide what to present to you today, I decided something unusual for myself, and that is to not talk about many mouse models that we're working on. We have several of them here as posters, which we'd love to discuss with you, including myself and the guys in the lab at the lunch break. But rather, I thought what I'd do is follow on from the presentation that Francesca gave you yesterday, and tell you about an example of how we can use humans as a beautiful model organism to help us understand mouse cancer. And so there's two messages I'd like to give you here, and for anyone who wants to check their emails for the next 13 minutes can listen now. We think sex-specific differences are something that shouldn't be ignored, and we think that KDM5C and KDM5D are actually not the same. So to come back to what Francesca was telling yesterday, obviously it's very well characterized that in sporadic clear cell renal cell carcinoma, there's a very strong incidence bias. Men get cancers more often than women. And as I was preparing this presentation, I thought I'll just write a little line saying that it's the same in VHL patients with CCICC. And I started Googling and looking, and actually it turns out I couldn't find anything. And maybe the clinical colleagues can correct me here, but I think it's not really clear whether there are in fact differences in CCICC incidence in male and females in VHL patients. And I wrote a quick email to Atina Garner, the colleague from Freiburg who's actually here, in the audience, and asked her this question. She said she didn't know, but she looked in the Freiburg registry of VHL patients, as shown in this plot here, and characterized only those VHL mutations that were defined as being loss of function in the recent Nature Genetics paper screening for different functional variants. And what she saw was sent me back within about half an hour this nice graph that shows that, at least for this subset of patients, there is actually a difference. And I think this is maybe to my mind, an unanswered question, and we'd love to talk to people in the field who maybe have similar data that we could think about. So I think there is an intrinsic difference in the biology of tumors between males and females. And ignoring now for the moment any hormonal differences and physiological differences, as Francesca said, what's clear is that there are genetic differences, most strikingly that KDM5C is mutated in male CCICCs, and as she said yesterday, that 5C in females, in women, there are two active copies of KDM5C. In males, there's one copy of KDM5C and one of 5D. And it's thought in general that KDM5D functions as a surrogate KDM5C in men. This is because of the very high amino acid homology and the fact that both of them have this same biochemical capacity to change or demethylate the trimethylated mark on histone H3 lysine 4 to a monomethylated. And by acting directly at promoters and enhancers and changing the balance of mono and tri, it's thought that the KDM5s actually thereby regulate gene expression. Two other little important things. We know from many talks yesterday that KDM5C is not a truncal mutation but arises later. We see that, for example, in higher frequency subclonally and in metastases. KDM5C also, along those lines, can be found together with many different CCICC mutations, so most prominently with polybromo and SETD2, actually never with BAP1, but we also find it with P53 and mTOR and P10. So it seems to cooperate probably in different ways with different mutations. Another interesting thing is that when we look across all tumor types, KDM5C mutations show the typical pattern that one might expect of a tumor suppressor. The mutations are scattered throughout the entire length of the protein. The black ones here are quite striking. They're truncating mutations that are predicted to cause complete loss of function. And what's interesting in kidney cancers that these are more prevalent, these truncating mutations, in about 2/3 of the cases. So there seems to be quite a strong selection for really killing KDM5 function. So that's what we decided to do in human CCICC cell lines. We designed sgRNAs to inhibit or to mutate KDM5C in exon 8, and then put these in a lentiviral using lentiviral CRISPR into populations of CCICC cells that we know through exome sequencing are actually KDM5C wild type. These are the 4 cells we've been working on here. You see KDM5C is nicely expressed, and again, when we generate these populations of cells in each of the different cell lines, we can nicely get rid of KDM5C protein expression. We deliberately work with whole populations of cells here because we want to avoid any potential clonal artifacts that can come from selecting out individual clones. What we see now in all of these pictures, all of these diagrams, you'll see the control cells in gray, the 2 knockout clones in blue. We basically see no difference in proliferation in normal cell culture. There's a very small difference in the 2 female cells in terms of increasing colony formation. This is not true in the male cell line where KDM5D may be compensating, and I'll show you a little bit of data about that in a minute. We see differences in xenograft assays. So in 7860, there's no effect. In CACI2, there's actually a blocking or a slowing of tumor formation. There's an acceleration in A498. And 769P cells, which don't grow as tumors, normally also don't grow as tumors when we've knocked out KDM5C. So we see different kind of phenotypic effects, albeit fairly mild ones, of KDM5C deletion or mutation in these cells. Transcriptionally, we also see actually relatively mild effects. So there are very few genes whose expression is changed in the knockouts. And the interesting thing is that there's basically no overlap in the upregulated or downregulated genes between the different cell lines. And I think this is consistent with the fact that KDM5C is not really a primary transcriptional regulator, but is probably recruited to different sites depending on the underlying state of the chromatin that's governed by many other factors. And consistent with that, when we localize KDM5C peaks in A498 or 7860 cells with cut and run, you see that there is a small overlap, but largely the binding patterns are quite distinct. So it's probably that KDM5C has cell type specific functions. We can confirm biochemically at least that KDM5C is doing what we think it should be doing. So when you do cut and run for all of the KDM5C binding sites, you can see that it is enriched in sites that have monomethylation, which is the mark that's actually kind of written by the demethylase activity of KDM5C. Some of these are probably active promoters because they're high in trimethyl. And these sites are actually reduced, as we might expect, in the knockouts. What's kind of interesting is that when you just look at all of the sites across the whole genome of monomethylation, we see that globally these are actually reduced as well in the knockouts, while KDM5C is not actually binding there. So either the cut and run antibody is not fully efficient, or these are sites that don't kind of require ongoing KDM5 activity. Now you might expect that the differentially expressed genes should be enriched for binding of KDM5C, either at their promoters or enhancers based on the common model. And what this diagram shows you that that's actually not the case, surprisingly. So when we take in A498 all of the upregulated genes and look for promoters or enhancers or the downregulated genes, we don't see any enrichment for KDM5C binding, but we do see this sort of global effect on monomethylation. So we can't really say actually that KDM5C is directly regulating these genes. Now, of course, you can't talk about KDM5C in males without talking about Y chromosome loss, where KDM5D is located. And the kidney, in general, kidney tumors actually show quite a high incidence of loss of Y chromosomes, as shown in this Cell paper. It's quite striking in papillary renal carcinoma, but in CCRCC it's about 40% of cases. What's not known is how this correlates with KDM5C mutation, and you can do that by simply looking at the expression of Y chromosome encoded genes. They basically all show the same pattern. What you see in the KDM5C wild type is that there are many tumors that have very low expression of Y chromosome genes compared to normal. We're only looking at male tumors now. And this number happens to correspond to more or less the 40% that has been shown by fish. What's very interesting is that almost all of the KDM5C mutant tumors have also lost KDM5D. So this is a state we decided to model, which I'll come to in a second. What I want to show you is that this Y chromosome loss, just a small aside, we know that in the mouse model of VHL-P53RB deletion, we also see this gender bias, or the sex-specific bias, I should say, in mice, that male mice get tumors earlier than female mice, they get more tumors. And what's interesting is that they also show this pattern of about 35% in this case of loss of the Y chromosome, apparently. So this is something that seems to be conserved across species and across cancer models. We therefore decided to now knock out KDM5D on top of KDM5C in the male cell lines, and we chose 7860, which has kept the Y chromosome, it expresses KDM5D. Here we knocked out 5C, here 5D, and here the double knockout. And what you see again is basically no change in proliferation. Now we see actually this little increase in soft agar colony assay formation. But the reason I really want to show you this data is this surprising observation. When we put this into xenograft setting, as I showed you before, KDM5C has no effect. KDM5D surprisingly completely killed tumor formation. And this we could fully rescue by deleting KDM5C and 5D. So this clearly shows that 5C and 5D are not just siblings that do exactly the same thing. They're not compensating for one another. In fact, they're sort of antagonistic in that respect. So we see a similar thing when you simply look at the tumors. So 7860 generally grow with this sort of mix of squamous and clear cell morphology. That's the same in 5C knockout. The 5D remnants of tumors look very more benign, and this appearance was rescued by the double knockout. There's a non-specific trend to decreased metastasis spontaneously from these subcutaneous tumors in 5C knockout, and again, this seems to be a little bit rescued by the double knockout. We see the same thing at the transcriptional level, that there are differing effects. So the genes that are upregulated in 5C or 5D are both distinct as well as overlapping. So they may have both overlapping and distinct transcriptional functions. And one can see very nicely the dominance of KDM5C over 5D when you simply look at those genes that are 5C dependent, shown here. They're obviously not affected by KDM5D. And if you knock out both again, you restore this, or you keep this KDM5C pattern. But the opposite situation is when we have the 5D specific genes, they're actually rescued by KDM5C co-deletion. And what's interesting is that many of these upregulated genes are putative tumor suppressors. So that may explain this effect that we're seeing. Again, when one tries to look at similarities and differences between 5C and 5D, we see there's a little bit of an overlap in binding sites. There certainly seem to be binding to different sites, although I would caution here using two different antibodies. We don't know about their relative efficiencies in cut and run, so it's a little bit hard to discuss there. And without going through all of the data, we see exactly the same thing. The differentially expressed genes are not preferentially bound by 5C or 5D. So this speaks against this common model that is proposed in the literature. So this already brings me to the conclusion, and the take-home messages are that KDM5D is not simply the equivalent homologue of KDM5C on the X chromosome in females. We don't know biochemically how they're different. That's something we're really working on. But we do know that, at least functionally, when we mutate KDM5D, we have a KDM5C dependent change in tumor formation as well as gene expression alterations. Now, this is a working model, and I'm drawing a very long bow when I try and claim this, but it's at least giving rise to these ideas that we hadn't really thought about before. And that is that in most cases, at least in men of CCICC, which are driven by all of the gene mutations we've been talking about, the X and Y chromosome are perfectly fine. So that's about 60% of cases, but in 40% of male cases, an event happens that causes loss of the Y chromosome. And if we assume for a moment that, as we hypothesize, that KDM5D mutation is the same as Y chromosome loss, we can't claim that, and that's what we're testing. One might expect that this may actually act as a brake or a proliferative block on the formation of tumors. And we can speculate that one way of overcoming this is for the cells to select for mutation of KDM5C, which would then allow tumor formation to continue. But what's also interesting is that there are many tumors where KDM5C is wild type that have lost the Y chromosome, and something else has probably happened, if this model is true, that has let them get around this barrier. And we don't know what that is. It doesn't correlate with anything sort of commonly that we might think. So that's something we're trying to understand, and I think it would be really wonderful to talk again to all of the people who showed the beautiful work that they've done looking at evolution of CCICC to see if we can tease out a little bit more collaboratively maybe when 5C and mutation occurs, when Y chromosome loss occurs. I think that would be a very interesting question that we'd love to discuss with you further. And the final take-home message is a more general one and kind of an appeal to everyone, and that is that in classical art we would never confuse David and Venus as shown here. And I think in medicine in general, there's an obvious realization that men and women behave differently, but in the research context, in clear cell renal cell carcinoma, in VHL disease, I think we have to be very aware that there's very different physiology, there's very different genetics between males and females, and it's really important to think about gender differences when we're looking at all of these things. And something that's actually, I must say, is not really reported on. There are many mouse studies, for example, where it's not even mentioned what sex the mice were. That's something that has to be improved upon. We have several examples. Basically, in all our mouse models, we always split the genders, and as I said, we're very happy to discuss some of these interesting gender-specific or sex-specific differences in different mouse models that we have. And as a second aside, I'd love to talk to all of the kidney cyst people, specifically at this poster, maybe if you have time in lunch, where we have another story that I actually would have liked to have presented today. So with 20 seconds left, I'll close it here. I thank you for indulging me, and thank all of the wonderful people that I really have the privilege working with in the laboratory. Thank you very much.
So I'm just wondering, did you check the KDM5C and 5D in terms of appliance knockouts effects on the H3K4 translation? either on the protein level or on the ChIP-seq level?
Yeah, so in A498, we very clearly see that there's at least at the ChIP-seq level, there's an effect. What's interesting is in 7860 cells, neither the single nor the double knockouts have a global effect like we see in KDM, in A498. We don't know in any other cell lines. It may relate to possible compensation by KDM5A and 5B and different effects there, but we can't answer that question. It's a good question. Yeah.
Thanks.
Thanks, Ian. That's fantastic. To what degree is this PBRM1 dependent? Do you have a sense of whether that background is important in influencing this interplay with KDM5C and D, or is it?
We don't have any indication of that. You know, the 7860 model is polybromo wild type. It has P10 and P53 mutations. You know, a cell line study of n equals 1 is very hard to conclude anything rigorously from that. We think we have some preliminary evidence from other studies we've been doing that knocking out polybromo may actually, as Rui showed and others have showed, may increase a tendency for aneuploidy and chromosomal instability and select actually for Y chromosome loss. That's something we're working on now. This interplay between the sort of these driver mutations then selecting for specific chromosomal abnormalities.
And in the P53 mutant mouse model where you see worse outcomes in male animals, is that correlated with the third where you see Y chromosome losses in some way?
No, we don't. We don't have the resolution to ask. That's a good question. Yeah. Exactly. Is Y promoting or inhibiting tumor formation? Yeah. We can't. No, I don't have that. Yeah, it's a good question. Yeah.
Thank you, Jan. Wonderful presentation. Also from all the molecular aspects. I would also give you a provocative point of view from a clinician that probably is less fancy regarding what you've shown, but the difficulty in treatment is completely different. So to operate on female, it's much easier in terms of surgery, in terms of less toxic fat, less BMI, less comorbidities. For a clinician to give an indication of an active treatment in VHL patient, female versus male is a completely different topic.
That's very interesting. Thank you.
Thank you so much, Ian.
Thank you very much.
Super cool.
So that concludes this session. I want to thank all the speakers for the wonderful lectures they gave and the audience for being very engaging and stimulating a wonderful discussion.